Hi, my name is Cleo Schied, and for my science experiment, I chose to do prosthetic plant tissues. I was inspired to do this experiment after I discovered a couple studies done by Vanderbilt University and another group of researchers who actually turned cotton candy and spinach into a functioning human capillary network and even a beating human heart, as seen in these pictures. For my process, I tried to follow their process as much as I could, although not a lot of their details were released into the decellularization process in order to use perfusion to make the cells transparent in order to see the mold go into the plant to make a capillary structure. So in order to do this, I soaked the specimen, in a bleach solution for 10 days. And after the 10th day, you could see that there was some results of different transparency in the structure, but I could not ultimately get it all the way clear. I did end up having the specimen end up rotting after a few days, so in order to make up for lost time, I ended up just injecting the gelatin mold into the specimen directly. And as you can see, those results are up here. Initially, I saw a bit of reddish tint into the plant, but I ultimately didn't think it was going to be effective. And so in this case, I kind of just was like, okay, we'll dry it out and see what happens. And a few days later, I came back, and it turned out the gelatin actually did form into the capillary network of the plants, which shows the vascular structure. As you can see, the areas where the gelatin was effective, the plant around it actually rotted away, yet where the mold held up, the plant held up as well, which I thought was really cool, and I was not expecting that at all. I think plants are actually going to be the future of regenerative medicine for plant tissues because they have a very high surface area, they have excellent water transportation and retention, and they can mimic the human capillary network very well and are a very good replacement for future prosthetic organs. And as shown with studies by Vanderbilt University and with the researchers by John Gluskick, who did the beating human heart tissue, I think it's very possible that we can use these plants to mimic human capillary networks and soon use plants to be a good source of prosthetic organs in the future. So thank you very much.